Hello, everyone. My name is Feng. I'm a postdoc from Bingren's lab. First, I want to thank the organizers for giving me this opportunity to present our work here. This is a great conference. So for the past few years, we have been working on a project to reconstruct a map of cis-regulatory elements in the mouse genome. So the goal for this project is really just to address the following three simple questions. Where are the locations of the cis-regulatory elements in the mouse genome? And what transcription factors are involved in regulating tissue-specific gene expression? And how do we link enhancers to their target genes? These are the three classes of regulatory elements we are interested. In the center, we have the promoter sequence. This is where the RNA polymerase binds and initiates the gene transcription. On the right side, we have enhancer, which generally serves the hub for the transcription factor binding sites. The enhancer can interact with the promoters through chromatin looping, and then thus enhance the gene transcription. On the left side, we have insulator, which is generally bound by CDCF. The classic view of insulator is that it can block the enhancer interacting with the wrong promoter. So to map these cis-regulatory elements in the mouse genome, we performed ChIP-seq in 19 mouse tissues, including 13 adult tissues, 4 embryonic tissues, and 2 cell lines. For promoters, we use H3K4 trimethylene mark and PAL2. For enhancers, we use H3K4 monomethylation. And for active enhancers, we use H3K27 acylation, as discovered by our speaker, John Wasaka's group. And for insulator, we use CTCF. So from the 19 tissues we studied in this project, we were able to identify over 50,000 promoters, 100,000 CTCF binding sites, and over 200,000 enhancer sequences. So putting everything together, we have this map of non-redundant, nearly 300,000 regulatory elements in the mouse genome, and they cover about 11% of mouse genome. This is a great expansion of the annotation of the mouse genome, compared with the 2% of the protein coding sequences. Next, to determine the accuracy of our promoter or enhancer prediction based on chromatin features, we performed some experiments. So we randomly choose 65 promoters and 8 random enhancer sequences, and we performed the luciferase reporter assay. And we are very pleased to see that over 85% of the promoters and 75% of enhancers show strong and significant promoter activity or enhancer activity, supporting their role as a promoter or enhancer. Next, we compare the cis-regulatory elements with conserved non-coding sequences. Comparative genomics has identified a large number of sequences that are conserved during the course of evolution, but not used for protein coding. They are suspected to have significant biological roles, but their exact functions were not clearly defined. So in this study, we first downloaded this list of 1.4 million conserved non-coding sequences and compared them with the cis-regulatory elements. And we found that over 70% of the non-coding sequences, they fall with at least one class of the regulatory elements, 15% as promoters, 53% as enhancers, and 2% of the CTCF binding sites. So it seems that the majority of the conserved non-coding sequences, they have biological functions in gene regulation. Mammalian development require a precise spatial and temporal gene expression program that is tightly controlled by transcription factors. So this map of cis-regulatory elements provides us this excellent opportunity to identify systematically the transcription factors involved in tissue-specific gene expression. To do this, we first identified 19 clusters of tissue-specific enhancers. And in this, as shown in this heat map, each column is a tissue or cell type, and each row is an enhancer. So we performed some gene ontology analysis. The results confirmed that the tissue-specific enhancers, they are located near the tissue-specific expressed genes. For example, the mouse ES-specific enhancers, they are located near the genes important for stem cell differentiation or stem cell maintenance. And embryonic brain-specific enhancers, they are located near the genes that are important for brain development. Next, we performed the motif analysis for these tissue-specific enhancers, and we are very pleased to find that stem cell-specific transcription factor OCT4 is only enriched in mouse ES-specific enhancers, and HNF1 is only enriched in kidney or liver-specific enhancers. And what is more interesting is that the motifs we found inside these enhancer regions, they are actually evolutionarily conserved. And this gives us another layer of confidence that potentially these transcription factors might be important regulating the tissue-specific gene expression. So in total, we were able to identify nearly 270 transcription factor motifs. And as shown in this heat map, again, each column is a tissue or cell type, and each row here is a transcription factor motif. The color here indicates the significance of enrichment. Yellow is more enriched, black is less enriched. So you can tell that the tissue-specific enhancers, they are really enriched for tissue-specific transcription factor motifs. For example, this cluster of mouse ES-specific enhancers, they are enriched for OCT4, SOX2, and the EKL4 motif. And this cluster of tissue-specific enhancers, they are bone marrow and spleen-specific. They are enriched for PU1 and ETS and AML motifs. And most of these motifs, they're actually evolutionarily conserved. So far, I have introduced our strategy to map the locations of the cis-regulatory elements in the mouse genome, and how do we define tissue-specific enhancers, and how do we found the transcription motif factors. And next, we want to describe our efforts to how to link enhancers to their target genes. So enhancers, they are really the primary driver of the tissue-specific gene expression, but linking enhancers to their target genes is not straightforward, because enhancers generally located distal to the gene they regulate. For example, in this case, we have enhancer located between two genes. So which gene does this enhancer regulate? We can simply assign this enhancer to the nearest TSS, as most literatures do. But we know that that's not optimal, and there's space for improvement. So in this study, because we have profiled the Crompton state for the enhancers and promoters across 19 tissues, then we can actually take advantages of their tissue specificity and link the promoters to the enhancer. For example, here, this enhancer and gene number 2, they are both mouse EL specific, and they are highly correlated. But this gene number 1 is heart specific, thus it doesn't correlate well with the enhancer. As a result, this enhancer will be assigned to gene number 2, although it is closer to gene number 1 in genomic distance. Performing this correlation analysis genome-wide, we were able to construct a heat map like this. So what we have here is a 1 million base pair region on the chromosome 19 in mouse genome. Each row or column here is either enhancer or promoter, and each dot in the figure indicates the correlation coefficiency. The red means positively correlated, and green is negatively correlated. So immediately we can find that the enhancers and promoters, they can form this kind of clusters, and we think these clusters are functional domains of co-regulated elements. Performing the analysis genome-wide, we were able to identify over 8,000 EPU and EPUs. The EPUs, we think they are the functional domains, and that the average domain size is about 140 kb, and the average enhancer to promoter ratio is almost 6 to 1, consistent with the idea that each gene can be regulated by multiple enhancers. So performing the EPU analysis genome-wide, we were able to link thousands of enhancers to promoters in each tissue studied in this work. Of course, we need to perform more experiments to verify some of the enhancer-promoter linkage. We first performed a series of 3C experiments pioneered by Job Decker's group. So in this region, we have an enhancer here marked by the green bar. So this enhancer is highly active in the mouse cortex, but it's silenced in mouse ES cells. And this is a promoter which is predicted to be regulated by this enhancer. So if what the EPU analysis is correct, then you would expect to see a strong physical interactions between these promoters to these enhancers. And that's actually exactly what we see here from the 3C experiments. So in this experiment, we use enhancer as an anchor point, and we design a series of primers around this promoter. And this sharp peak right on top of the promoter indicates that this gene strongly interacts with this enhancer. So in total, we performed 7 3C experiments, all confirmed the linked promoter and enhancers. To validate the enhancer-promoter linkage genome-wide, we performed high-C experiments in mouse cortex. And what you have here, the y-axis is the normalized high-C interaction frequency, and x is the genomic distance between two elements. The blue line here is the high-C interaction frequency between linked promoters and enhancers. The red lines are between promoters and enhancers are not linked by EPU analysis, and the green is random control. So based on this figure, you can tell that the linked promoters and enhancers by EPU show much stronger long-range interactions, which shows that our EPU analysis did a good job linking promoters to their distal promoters. Finally, the discovery of EPU actually also suggests that the mammalian genome is partitioned into functions of co-regulated elements. As shown in this figure, we have 3 gene clusters, PCDH alpha, beta, and gamma, our EPU analysis correctly identified 3 domains, domain number 1, 2, 3, and each domain corresponding to 1 gene cluster. And if we put the high C signals on top of this track, you can actually see some interesting observations. This is how to read this high C interaction heat map. So each data point indicates the interaction between this point and this loci. And the color of this point indicates how strong the interaction is. So you can actually, for this domain number 1 and number 3, we have these strong physical chromatin interactions just within the domains, but not between the domains. We have another example here where we identified a large functional domains of co-regulated enhancers or promoters. It covers 400 kb of the mouse region. And again, you can see these strong chromatin interactions just within this chromatin domain. And actually, this is quite similar to some observations made earlier this year. There are 2 back-to-back papers published in Nature. One is actually from our group. One is from Jaap Dekker's group and Edith Herth's group. That human genome and mouse genomes, they are organized in topological domains. And we think there's a strong correlation between the topological domains and the functional domains defined by cis-regulatory elements. So to summarize, we were able to identify nearly 300,000 cis-regulatory elements, covering about 11% of the mouse genome and 70% of the conserved non-coding sequences. We were able to identify the tissue-specific enhancers, and we identified nearly 270 motifs enriched in tissue-specific enhancers. And most of the motifs are evolutionarily conserved. And finally, we identified thousands of linked promoters in the enhancers through EPU analysis, and we found strong correlation between the functional domains defined by the cis-regulatory elements and the topological domains defined by the high-C experiments. This work was recently published in Nature, and we also built a support website where you can go and download all the raw data or the predicted elements. And you're also welcome to email me, and I can answer your questions. With that, I'd like to thank my advisor, Dr. Bing Yuan, for all the help, and my collaborator, Dr. Yin Shen, and our technician, David McCleary. They actually did all the experiments in this work. With that, I thank the audience, and I can take some questions.
Thank you for a very nice talk. I have 2 questions. So the first one is a very detailed question. On one of your slides, you basically showed that you may not have to go back to it, but when you were showing the 2 possibilities for linking, where you had a distal enhancer on the right and then 2 promoters on the left, certainly the shape matched more with the left promoter, but the total intensity was higher on the right promoter. So is it possible that there's a cryptic promoter that's not annotated, that it's interacting more strongly?
You mean here?
Yes.
Yeah, actually, we don't know why there's a peak right here, but actually the promoter is actually dip in the valley here.
Yes,
so either maybe that's because they're closer to the anchor, or maybe there's a novel elements we didn't identify, so we don't really know.
Okay.
Yeah.
So basically, these are the raw intensity signals. These are not corrected by distance.
No, this is 3C experiments. They are not. They has nothing to do with genomic.
Perfect.
Yeah.
That could be an explanation.
Yeah.
My second question is, you have this concept of an EPU in a way assumes that they're not crossing each other, but it is very well possible that many of these long-range interactions are in fact crossing. So. Yeah. Have you? Exactly.
Yeah, actually, that's a very excellent question. So to be honest, I think the EPUs will only cover, I think, 10 to 20% of all the enhancers we identified. So of course, EPU, you can see they are focused on the local interactions. Of course, there will be some long-range interactions that will not be captured by the EPU analysis.
So what you're saying is that basically about 20% of them fall into this unit concept.
Yes.
And the other 80% are...
Those are the elements we have confidence because they have the support by the tissue specificity. But there's other things we couldn't address at this point.
Great. Thank you very much.
Thanks.
One last quick question.
I'm mainly here looking for reassurance that what we see is not a result of a problem in our data. How often do you see clusters of enhancers that are tissue-specific, but where you can't find the tissue-specific output, tissue-specific gene expression level to correlate with them?
You were saying that when you see tissue-specific enhancer, but you don't see nearby genes that are tissue-specific expressed?
Exactly, or really a cluster of enhancers where you truly expect to see some output, but they're just not there.
That's a good question. Actually, we never looked into that, actually. But that's a good question. I think I can take a closer look in the future. Thanks.
So we actually need to move on. I apologize. And